Hi guys, I hope you're all well. So today we are talking about cholinergic and adrenergic receptors as well as their transmission. So by the end of this video, I hope you'll be able to describe the different types of cholinergic receptors as well as the different types of adrenergic receptors. Also, I hope by the end of this, you'll be able to describe the transmission of acetylcholine and norepinephrine from the axon terminal to their receptors. Okay, so let's start off by talking about the different types of cholinergic receptors. So I literally just took this, I took a picture of this diagram that I drew from my notes. So there are two main types of cholinergic receptors. So the first one is called nicotinic, which is an ionotropic receptor. It means it's just activated by ions. So they're actually ligand-gated ion channels. And the second type is called muscarinic. So muscarinic receptors are all metabotropic. So all muscarinic receptors of this type are G-protein coupled receptors. So if you don't know what G-coupled protein receptors are and the different types of G-protein coupled receptors, I have a video on my channel as well describing these. So they're basically the GQ, GI, and GS. So if you have no idea what I'm talking about, please watch that video first, because this will all make so much more sense if you know what I'm talking about. Right, so nicotinic receptors. Nicotinic receptors are sensitive to obviously acetylcholine as well as nicotine. There are two subtypes, so we have NN and NM. NN, they're mainly found in the ANS ganglia, but also in the central nervous system. And M, on the other hand, they're mainly found in the neuromuscular junction of skeletal muscle cells. So the muscarinic subtype, there are three different, actually there are five. So there's M1, M2, M3, 4, and 5. But for the purpose of this video, we're just going to stick to M1 to M3 because the other ones are not so relevant. So M1, so the first muscarinic receptor type is M1, and it's G-protein coupled receptor, as I mentioned, and it's coupled with GQ. So that means that the second messenger is IP3, DAG, and PLC, which ultimately results to the increase in calcium in the cell. So M1 receptors are mainly found in nerve endings, parietal cells, as well as some of the glands like the gastric and the salivary glands. M2, the second type of muscarinic receptor, it's coupled with GI. So GI, it's an inhibitory G protein. So the second messenger would be CAMP and PKA. So these receptors are mainly found in the heart muscle, specifically in the sinoatrial node, the atrioventricular node, as well as in the bundle of His. They're also found in some nerve terminals. M3, so the third type of muscarinic receptor, it's coupled with GQ as well, similar to M1. They are mainly found in exocrine glands like gastric, salivary, lacrimal, and other exocrine glands. They're found in the eyes as well. So there are two main muscles that we find this in the eyes actually. So the first one is the constrictor pupillae, which constrict the pupils, leading to miosis. The second muscle where we find this are the muscles connected to the lens. So those are called the ciliary muscles. Sorry, I was trying to remember. So yeah, the ciliary muscles and the constrictor pupillae. And then they're also found in the smooth muscles of the bronchi, the bladder, and the GI tract. They're also found in the beta cells of the pancreas, and they're actually involved in the release of insulin. So moving on to the adrenergic receptors, we have two main types. So we have the alpha adrenergic receptor and the beta adrenergic receptors. So the alpha adrenergic receptors are subdivided into alpha 1 and alpha 2. Alpha 1 is again all of these actually are G-protein coupled receptors as well. So alpha 1 is coupled with GQ. So again, that leads to the increase in calcium inside the cell. which means, either contraction or secretion. So they are mainly, alpha-1 receptors are mainly found in the smooth muscles of the vasculature. They actually control the resistance of the arterioles and venous capacitance, which ultimately lead to the control of the blood pressure. They're also found in some glands like gastric and salivary. So alpha-2, it's coupled with GI, so G inhibitory. Second messenger, again, CAMP and PKA. So they are mainly found, again, in the smooth muscles of blood vessels and in some nerve terminals. So moving on to the beta adrenergic receptor, there are three main subtypes, so beta 1, 2, and 3. Beta 1 is coupled with GS. So beta 1, they're mainly found in the cardiac muscles and the juxtaglomerular cells of the kidneys. Beta 2, again found in the heart, as well as the liver and some smooth muscles like the GI tract, the bladder, and the bronchi, more specifically. So the last one is beta 3. Beta 3 is coupled with GS. So all of the beta adrenergic receptors are GS, G stimulatory. So the second messenger would be CAMP and PKA as well, but they are stimulatory in contrast with alpha-2, which is inhibitory in nature. So beta 3 are mainly found in adipose cells. That's why they are involved in lipolysis if they're activated. Also, I forgot to mention on the previous slide when I was talking about muscarinic receptors. So muscarinic receptors also respond to muscarine as well as acetylcholine. That's why they're called muscarinic. And nicotinic, as I said, I think they respond to both acetylcholine and nicotine. Right, so now moving on to the really fun part, we're going to discuss the cholinergic transmission, and on the next slide, the adrenergic transmission. So we're going to talk about the synthesis, the storage, the release of these neurotransmitters, as well as the termination of their action. So let's start off with the synthesis of acetylcholine. So acetylcholine is made from choline and acetyl coenzyme A. So the first step is the transport of choline into the axon terminal. So this is the axon terminal, right? So the transport of choline into the axon terminal. So this is actually the rate limiting step of the whole reaction. So and this step can be inhibited by hemicholinium. Okay, so once again, choline is transported into the axon terminal and combined with acetyl coenzyme A via the action of an enzyme called choline acetyltransferase, and that makes acetylcholine. And this transport again is inhibited by hemicholinium, and the transport of choline into the cell is the rate limiting step in this reaction. Okay, so just to give you guys just to let you guys know, the colors, they mean something. So the orange writing, so that and that and that, so they are the important enzymes or like proteins that we have to know, and the writing that are like in red are the ones that inhibit a specific reaction. And if you see a writing that's like in green, then that means that promotes that kind of action or that process. Okay, so anyway, let's go back to acetylcholine. So acetylcholine is now made, and then the next step is for acetylcholine to be stored in a vesicle. And this storage process is facilitated by VAT. So VAT is vesicle-associated transporter. Okay, it's just basically placing acetylcholine into a vesicle for storage. Okay, this again can be inhibited by a drug called visamicol. Okay, so when acetylcholine is packaged in the vesicle, the influx of calcium helps with the exocytosis of acetylcholine. The exocytosis of acetylcholine, the release of this, can be inhibited by botulinum toxins. Okay, so when our acetylcholine, our little guy here, gets outside of the axon terminal into the synaptic cleft, two things can happen. So acetylcholine can bind to the cholinergic receptor right here. It could be like on another neuron, for example, and trigger a cascade of reaction there, or it can be broken down by acetylcholine esterase. So it's an enzyme that splits acetylcholine to choline and acetate. So this is kind of how the action of acetylcholine is terminated. And then choline and acetate are both recycled back into the axon terminal, and kind of the whole process starts again. So let's now discuss the transmission of noradrenaline from the axon terminal to its receptor. So noradrenaline, also called norepinephrine and epinephrine, are the main transmitters at sympathetic postganglionic neurons, with the exceptions of sweat glands, which release acetylcholine. So the first step includes the transport of tyrosine into the axon terminal. So when tyrosine gets into the axon terminal, it gets converted into a compound called dopa. So this reaction is facilitated by an enzyme called tyrosine hydroxylase. Okay, so tyrosine is hydroxylated to dopa via tyrosine hydroxylase, and this reaction can be inhibited by a drug called metyrosine. So once dopa is synthesized, it is then decarboxylated into dopamine. So when we have our dopamine, again, we need to store dopamine in a vesicle, and this storage is done by VMAT, vesicular monoamine transporter. The storage of dopamine into these vesicles can be inhibited by a drug called reserpine. Okay? And then once our dopamine is packaged into the vesicle, it gets converted into norepinephrine. And then, again, the exocytosis of these neurotransmitters require calcium. So the influx of calcium promotes the exocytosis of norepinephrine. So the release of norepinephrine here can be inhibited by a drug called guanethidine, and can be promoted by these drugs called amphetamine and ephedrine. So when norepinephrine gets out into the synaptic cleft, three things can happen. So it can bind to the receptor right here. It could be on like an effector muscle or something, and then obviously trigger like a contraction there. It can also be broken down by this enzyme here called catechol-O-methyltransferase, or COMT for short. So that has that negative sign because it inhibits the action of norepinephrine by reducing its concentration in the synaptic cleft. So it gets broken down into an inactive metabolite called norepinephrine. So norepinephrine is excreted in the urine. And just a fun fact, actually, the inhibition of COMT is actually very useful in Parkinson's disease. So the last thing that can happen to our norepinephrine is it can be recycled back. So the reuptake of norepinephrine is facilitated by NET, so norepinephrine transporter. And this process can be inhibited by cocaine and tricyclic antidepressants. Also, there's this is a mitochondrion. So this is my best illustration of a mitochondrion. So inside mitochondria of the cells in an axon terminal, there are these chemicals. They are actually enzymes as well, called monoamine oxidase, MAO for short. So they are present inside mitochondria, and they are responsible for degrading catecholamines. For example, norepinephrine, epinephrine, serotonin, and dopamine. So they also inhibit norepinephrine and dopamine by reducing their concentration in the cytoplasm. So the degradation of these amine neurotransmitters in the cytoplasm via MAO produces inactive metabolites as well, which are then excreted through the urine as well. By inhibiting MAO, we can then increase the stores of these neurotransmitters in their nerve endings. So I hope that all made sense. Thank you again for watching my videos, and if you have any questions, please leave your question below and I'll try answer them as soon as I can. Thank you.